You know, inside your body, there's this constant drama unfolding. It's a story of how your cells respond to challenges, how they adapt to new environments, and sometimes how they take a really dangerous turn. So today, we're going to decode two key chapters of that story, metaplasia and dysplasia. To really get this, let's go back a bit. Think about your first day of school, right? Everything was new, everyone was kind of like a blank slate, and the possibilities just seemed endless. You and your classmates, you could have grown up to be anything. And believe it or not, your cells start out in a very similar way. It's actually surprisingly close to how our cells work. We all start out as these unspecialized stem cells. Think of them like a classroom full of new students. Then, guided by our genes and their environment, they go through this process called cellular differentiation. They become specialized cells. You know, heart cells, skin cells, lung cells, each with a super specific job, just like all your old classmates chose different careers. But what happens when that environment changes? What if our cells are put under constant pressure from, say, outside stressors? Well, that's when their normal developmental path can get totally thrown off track. And this, right here, is where our story really begins. So, the body's first response to this kind of chronic stress is often a really clever one. It's a protective adaptation that involves a smart switch to a tougher type of cell. And this whole process is called metaplasia. What's so fascinating about metaplasia is that it's a change from one fully mature, specialized cell type to another fully mature, specialized cell type. Think of it like a software engineer who retrains to become a construction manager because, well, the job site is a much harsher environment than an office. It's a very intentional, very organized switch. Let's look at a couple of classic examples to make this real. So in a smoker's lungs, the delicate column-shaped cells that line the airways just can't handle all the toxins in tobacco smoke. So what do? They change into these tougher, layered, more durable squamous cells that offer more protection. Or think about someone with chronic acid reflux. The cells in their esophagus aren't built to handle acid, so they transform into the same kind of acid-resistant columnar cells that you normally find in the intestine. It's an amazing adaptation. In this slide really breaks down that acid reflux example perfectly. So first, you've got the stressor, that chronic stomach acid. It starts damaging the normal esophagus cells. In response, the body's stem cells adapt and start producing these tough, acid-resistant columnar cells instead. This new, better-suited lining creates a condition we call Barrett's esophagus, and it is a perfect illustration of metaplasia in action. Now, here's the good news, and this is absolutely crucial. Metaplasia is reversible. If you take away the stressor, by treating the acid reflux or quitting smoking, the body can stop making those replacement cells, and the tissue can eventually go back to its original state. It's an adaptation, not a permanent sentence. But that raises a really critical question, doesn't it? What happens if that stress just keeps going, day in and day out, with no relief? Well, the body's organized, clever adaptation can start to break down, and that can lead to something far more concerning. And this brings us to dysplasia. And let me tell you, this isn't an orderly switch anymore. This is the beginning of cellular chaos. So dysplasia is the abnormal development and just wild growth of immature cells. Instead of that neat, orderly switch we saw before, the tissue starts filling up with this disorganized mix of irregularly shaped, underdeveloped cells that aren't even doing their jobs right. When a pathologist looks at this under a microscope, they're looking for 4 key signs of this chaos. They see cells of all different sizes and all kinds of weird, abnormal shapes. The cells also look really dark with too much pigmentation. And finally, they see way too many cells that are actively dividing. This chaotic picture is the hallmark of dysplasia. And yeah, that combination is just unequivocally bad news. This uncontrolled growth of abnormal cells is precisely what defines a precancerous state. It's the big, flashing warning sign that the cellular machinery has gone haywire. Let's see how this can progress, using cervical cancer as an example. The stressor is often an HPV infection, which can cause the cervical cells to become dysplastic. If that keeps going, it can become something called carcinoma in situ, which means the uncontrolled growth is still contained. But the truly dangerous step is when those cells break through their boundary, this thing called the basement membrane, and start to invade other tissues. That is invasive cancer. Okay, so now that we've seen both sides of this cellular story, let's put them side by side. Let's do a final comparison between that organized adaptation and this dangerous, uncontrolled chaos. This table really sums it up perfectly. Just look at the cell change. Metaplasia is a switch between two mature cell types. Dysplasia is just a ton of immature, abnormal ones. What about reversibility? For metaplasia, it's a clear yes if the stress goes away. For dysplasia, it's a maybe, and only if it's caught really early. And the most critical difference of all, the cancer risk. Metaplasia is benign, dysplasia is the definition of precancerous. So if you take just one thing away from this whole explainer, let it be this. Metaplasia is the body's organized, strategic adaptation to a problem. Dysplasia, on the other hand, is disorganized, chaotic, and uncontrolled growth that has completely lost its way. One is a solution, the other is the beginning of a much, much bigger problem. And that leaves us with a final thought, doesn't it? Our cells are constantly listening. They're always responding to the environment that we create for them through our choices. So the real question is, what signals are you sending? Are you asking your cells to adapt, or are you pushing them towards chaos?